Ouais, c'est fini. Bon, ça change pas grand-chose. Là, il y a des discussions qui sont en train d'être publiées. Organiser les c'est très sensible. Je pense que ça va être pas mal. Vous avez la charge. Oui, c'est ça. Prendre le c'est ça. Je pense que c'est pas mauvais choix. Je pense que c'est bon.
Okay, good morning everyone. So we're going to start. Welcome to the seminar. So as usual, we are on a webinar mode. So people online, do not hesitate to report any technical issue in the chat and to ask your question in the Q&A tab. And before starting, we just wanted to encourage all of you to submit suggestions for next winter speaker sessions for the seminar. As we at the moment do not have that many suggestions, with the little specificity for this call that because we are transitioning from LabX to another way of funding, which I forgot the name, we will have no funding for next winter for speakers, but that's the opportunity to suggest local speaker or colleagues that you know will come anyway for working on projects or anything. So we are waiting for suggestions. And with that, I thank Mathieu for introducing to the speaker.
Okay, good morning. It's my great pleasure to introduce Thomas Aubier. So Gila Ganem invited him, but I happened to be introducing him. He was my former PhD student, so he was working on the role of signaling and made choice in speciation and macroecology. And then he defended his thesis in 2018. Then he went on a 2-year postdoc with Hannah Coco in Zurich, working on sexual reproduction and red queen hypothesis, always on theory. So Thomas is a theoretician doing modeling, various ways of modeling things. Then he went on another postdoc with in the US with Maria Cervello, working on prezygotic barriers and recombination. And then he got recruited at CNRS in Toulouse, in the now what's called the CRBE, working on speciation and complementary arrangements, always from a theoretical point of view, I think. And he's going to give us probably a synthesis of all that.
Yeah.
Okay.
Thank you. So thank you for the introduction. Thank you both for having me here. So as Mathieu said, I am a theoretician and I am mostly working on evolutionary processes such as local adaptation and speciation. And today I'm going to present some studies I did on the coupling that occurs between reproductive barriers during speciation. Okay, so to begin with, let's ask ourselves this very simple question. So what does life look like? So you can first look around you, look at your colleagues. You can look at those other faces. So you're here looking at a small sample of humans. And even if there is some variation between individuals, you will agree that we are very similar to each other and that most of this variation is actually continuous. Now, if you consider life from a much broader perspective, then you will observe a greater variation. And this variation is mostly discontinuous so that we can classify living organisms into some kind of clusters that we call species. So to put it simply, if we reduce phenotype to just two phenotypic traits, here, color and size, if you consider life, you will not observe this kind of continuous variation. You will instead observe this kind of variation that is discontinuous and organized into clusters. So this is true for phenotypic variation, but also for genetic variation. If you do a principal component analysis on genetic data, you will get something like that. So life is discontinuous and organized into discrete clusters that we call species. And this raises another fundamental question, which is actually the motivation behind my work. How do these clusters form? So in other words, how do new species form? How does speciation occur? To tackle this question, we first need to agree on what we mean by species. So there are multiple definitions of species, each useful in their own way. But to study speciation, which is the process by which new species evolve, it is important to remember that it results from the gradual evolution of phenotypic traits. So it is a continuous process. And therefore, to study speciation, it is not important to be able to precisely delimit species. It is important instead to be able to use the definition of species within an evolutionary framework. And that's why most people studying speciation use the definition of species following the biological species concept. According to this definition, species are groups of interbreeding natural population that are reproductively isolated from other such groups. So this definition, as any definition, has many flaws. So in particular, this definition does not apply at all to living organisms that reproduce asexually, but it has the advantage of defining a research program, the study of the evolution of reproductive isolation. So in addition, defining species as groups of interbreeding natural population allows us to understand the generation and maintenance of this organization into clusters. So if interbreeding was occurring between species, then intermediate forms would be produced and we would lose this organization into clusters. This is not the case if there is reproductive isolation between species, as is often observed in nature. So to sum up, studying speciation is about studying the evolution of the reproductive barriers that lead to reproductive isolation between populations. And by reproductive barriers, I mean any processes that reduce gene flow between populations. So for instance, in this illustration, there is no reproductive isolation. So there is a cross between individuals from different populations. This leads to an offspring that is fully viable and fertile. So there is gene flow between populations. In these illustrations, there is reproductive isolation, and we can roughly classify reproductive barriers into two categories. Some barriers lead to prezygotic isolation, so that there is no formation of hybrid zygotes. So this is the case, for instance, when there is some mate choice that reduces crosses between individuals from different populations. And other barriers lead to post-zygotic isolation, so that hybrid genotypes associate with a low fitness. This is the case, for instance, when genetic incompatibilities have accumulated in the population, so that incompatible alleles are found together in hybrids and together increase mortality. So importantly, some barriers actually lead to both prezygotic and postzygotic isolation. This is the case of the local adaptation of population. So on the one hand, the local adaptation of population makes that individuals from different populations are less likely to meet and mate. So this is prezygotic isolation. On the other hand, the local adaptation of populations may reduce the average fitness of hybrid genotype. This is polyzygotic isolation. So of course, multiple reproductive barriers can occur at the same time, and some processes can even favor the coincidence of reproductive barriers. So this is what we call coupling. So those processes make that reproductive barriers operate together in such a way that reproductive isolation is increased. So this is the case, for instance, between those barriers I told you about. So between prezygotic isolation caused by mate choice and polyzygotic isolation caused by genetic incompatibilities. So the evolution of such mate choice that lead to prezygotic isolation is favored when the thinness of hybrid genotype is particularly low. This is the case when there is such genetic incompatibilities. And on the other hand, the accumulation of these genetic incompatibilities is favored when there is little gene flow between the population. This is the case when there is such mate choice that lead to prezygotic isolation. So those barriers are said to reinforce each other. And in the speciation literature, such coupling between barriers has received a lot of attention. And under this view, the establishment of strong reproductive isolation and therefore speciation relies on the accumulation of barriers. So the question I'm going to tackle during this presentation is this one. Does coupling between reproductive barriers necessarily increase reproductive isolation? So to tackle this question, I'm going to show you studies where I use proof of concept mathematical modeling. So to be sure we are all on the same page about this methodology, here is a very quick recap. So the goal of proof of concept model is to test the logic in verbal hypotheses that we formulate to explain why a certain phenomenon exists. And the idea behind this methodology is first to observe nature and build a verbal hypothesis, for instance, about the coupling that occurs between reproductive barriers. Then we can pick some simplifying assumption and build a mathematical model. And then by analyzing this model, we can evaluate this verbal hypothesis and perhaps propose a new research directions. So importantly, the model may be built to qualitatively describe some processes, and therefore the prediction do not need to interface directly with data to tell us something interesting. And also, the model may rely on a great deal of abstraction, and if you're not used to this methodology, you may have this impression. So this will be the case during this talk. So I'm going to show you studies where I use the population genetics theory, so that is based on extremely simplistic assumptions. So in particular, in some cases, there will be no genetic drift. I also don't consider plasticity at all. I only consider sexual reproduction. Complex traits will be encoded by single genetic loci. And I also consider extremely simplistic ecological contexts. I hope that I can convince you that even if these assumptions are extremely simplistic, the model prediction tells us something interesting and useful about speciation. So let's get back to the main question. Does coupling between reproductive barriers necessarily increase reproductive isolation? So here's a spoiler. The answer is no. So I'm going to show you three mechanisms by which the opposite outcome can occur. So the coincidence of reproductive barriers may, under some condition, reduce reproductive isolation. So this is what we can call negative coupling. And so I'm going to show you three mechanisms. So let's start with the first study. So it's actually a study that I did during my PhD with Mathieu. So when I was at the CEF. So I told you that mate choice can lead to prezygotic isolation. I'm going to show you now that prezygotic isolating barriers caused by female choice and caused by male choice can, in some condition, reduce reproductive isolation. So in the well-known cases of adaptive radiation, so we know that prezygotic isolation is caused by some kind of associative mate choice, so that the mating cues are under selection, so that polymorphism is maintained. So it has been shown in the cichlid fishes, but also in Heliconus butterflies. So here in this illustration, the color is the mating cue under selection, and you see that there it is used as a basis for associative mating, with red fishes preferring to mate with red fishes and blue fishes with blue fishes. So of course, it's much more difficult than that in cichlid fishes, but in any case, in both cichlid fishes and in Heliconus butterflies, it has been shown that such associative mate choice can be expressed by both females and males. So during my PhD, I studied what happens if associative mate choice can be expressed by both sexes, and I investigated how it could affect reproductive isolation. So associative mate choice either expressed in females or in males increase reproductive isolation. So therefore, I hypothesized that when selection favors such mutual associative mate choice, reproductive isolation would be further increased. And to evaluate this verbal hypothesis, I built a population genetics model. In this model, I considered 3 diploid genetic loci, so that are also diallelic, so with 2 alleles each. One locus is encoding an ecological trait. So it's a trait that is actually called a magic trait in the speciation literature, because I assume that it is under disruptive selection, as shown above, where you see that the intermediate heterozygous genotype associated with holophileness. But it's also used as a basis for associative mating, as represented here, so that individuals may prefer to mate with individuals that share the same ecological traits than their own. So such associative mate choice is actually controlled by 2 other loci that are either expressed in female or in male. So at those 2 loci, one allele is coding for the absence of mating preference, and the other allele is coding for such associative mate choice. So you can see here that in this model, through the evolution of either female associative mate choice or male associative mate choice or mutual associative mate choice, you can get some prezygotic isolation between those 2 subpopulations that are differentiated at the ecological locus. So I assume that there are some differences between males and females. So as is often done in those kind of models, I assume that all females have equal mating success. So because of this assumption, we can expect that female associative mate choice is favored, given that it reduces the risk of producing such maladapted offspring. And by contrast, I assume that males may differ in their mating success. And in particular, I assume that males expressing mate choice may lose mating opportunities by rejecting unpreferred females. So because of this assumption, male associative mate choice may be either beneficial or deleterious, depending on how costly is such male mate choice. So there will be a parameter that will control for this feature. I will get back to it later. So I'm not going to show you the mathematical equation, but to give you an idea of what is the mathematical model, I have a set of 27 equations that describes how the frequency of each of the 27 genotypes change over generations. So I assume that generations do not overlap, and during each generation, genotypic frequencies are changing due to variability selection, so that there is disruptive selection, as I showed you, but also due to mate choice, mating, and the production of zygotes. And during this last step, in some cases, I assume that there is some kind of random sampling in order to implement genetic drifts. And I'm going to show you results in deterministic simulation without genetic drift, and other results from stochastic simulation with genetic drift. So with this set of equations, I could track the frequency of the alleles that are encoding female and male choice, so especially when the stationary state is reached. And I also track the hybridization rate between those subpopulations that are differentiated at the ecological locus. So here's the first result. So here on the axis, I'm varying the value of the parameter I briefly told you about. So this is the percentage of lost mating opportunities for males that express associative mate choice. So basically, if this parameter is high enough, we expect associative mate choice in males to be costly. While if this parameter is low enough, then we expect male choice to be beneficial because it reduces the risk of producing maladapted offspring. So in this model, in deterministic simulation, allelic frequencies at the loci that are encoding mate choice either reach 0 or 1. So I'm not going to show you the frequency, but just the outcome. So basically, you can see that assortative mating is always favored in females, so as we can expect given the assumptions. But we also, and then we see that assortative mating is favored in males only if this parameter value is low enough, as we may expect. So basically, you have here two selection regimes depending on this parameter value. Either selection favors mutual assortative mate choice on the left, or selection favors only female assortative mate choice. So now I'm going to show you what is the hybridization rate at stationary equilibrium, depending on those two selection regimes, so depending on this parameter value. So first, in deterministic simulation, you can see that the hybridization rate is lower when selection favors mutual mate choice than when selection favors only female choice. So this is in line with the verbal hypothesis that I formulated. The situation is rather different in stochastic simulation with genetic drift. So I'm going to show you the average hybridization rate in stochastic simulation, where I assume that the population size is equal to 1,000. So you see that the situation is different. So you see that on the right, that when selection favors only female choice, there is no big difference between what is happening in stochastic and in deterministic simulation. So which means that genetic drift is not extremely high. But here, when selection favors mutual mate choice, you see that there is a strong deviation between the outcome in stochastic simulation and that in deterministic simulation. And what I found particularly interesting is where I put this arrow. You see that even if selection favors mutual mate choice, we get on average a higher hybridization rate than when selection favors only female choice. So reproductive isolation can be lower on average when those barriers coincide, so when selection favors both female choice and male choice. And given that this occurs only in stochastic simulation, we can already conclude that this is caused by the joint action of genetic drift and selection on mate choice. So to make you understand what is happening here, I'm going to show you the outcome of a single stochastic simulation. So for that given parameter value. So here we are in a condition where selection favors mutual mate choice. So on this left figure, I'm going to show you how the allelic frequencies are changing through time over the course of a single stochastic simulation. So you have here on the horizontal axis the frequency of the allele that is encoding female choice. So this is the frequency of choosy females. Here on the vertical axis, this is the frequency of the allele that is encoding male choice, so the frequency of choosy males. So initially, I assume that we are at the deterministic equilibrium with both females and males that express assortative mate choice. So you will see this point that will move. And in parallel, on the right figure, I will show you how the hybridization rate is changing over time over the course of the same simulation. So remember that we are in a condition where genetic drift is not extremely strong, so it didn't make any difference when selection favored only female choice. And here is what's happening. So you see here that genetic drift has a notable impact, and from time to time, it triggers some kind of coevolutionary dynamics that associates with a burst of hybridization. So I can show you again. So the impact of genetic drift, and from time to time, some kind of coevolutionary cyclings with burst of hybridization. So you see here that for this condition where selection favors mutual mate choice, reproductive isolation is highly unstable, and this explains why the hybridization rate is on average higher than when selection favors only female choice. So we could we did get more detail about the coevolution dynamics, but to make you understand what is happening, we don't need to look at those details. Just remember the last dating night you had in your life, then you know that the outcome of a dating night depends on your choice, so what you think about the other person, but it depends also a lot on that other person's choice, so what this other person is thinking about you. The same applies here when selection favors mutual mate choice. Selection on mate choice in one sex depends on mate choice in the other sex, which explains the coevolution dynamics that I just showed. So to conclude on this part, here I showed you that prezygotic isolating barriers caused by female choice and male choice can, in some conditions, reduce reproductive isolation. So this is some kind of negative coupling between those barriers that is driven by the interaction between selection and genetic drift. So now I'm going to show you another study that I did during my last postdoc in Chapel Hill with Maria Savidio. So I'm going to show you another mechanism by which the strengthening of a barrier can associate with the weakening of another. So this can occur through the antagonistic effect of a factor on reproductive barriers. And here I'm going to show you how the linkage between loci can favor local adaptation, which is a barrier, as I explained in the introduction, but inhibits the evolution of mate choice, leading to prezygotic isolation, which leads to an overall reduction in reproductive isolation. So in the previous study, I told you that I considered the magic traits. So magic traits are ecological traits that are subject to divergent or destructive selection and that are used as a basis for associative mating. So in the speciation literature, magic traits have received a lot of attention because they are traditionally thought to strongly promote reproductive isolation. So they have been shown in nature in some cases. For instance, in heliconous butterflies, the wing color pattern is an ecological trait under divergent and destructive selection due to differential predation, but it's also a mating cue used as a basis for associative mating. But in many cases, reproductive isolation may actually rely on distinct genetic loci that are encoding these different traits, ecological traits and mating cues. For instance, in the plant Mimulus, so it has been shown that the corolla characteristics and the shape of the leaves are ecological traits under divergent selection in context of local adaptation, while the flowering time is a kind of mating cue that strongly determines prezygotic isolation. In Mimulus, it has been shown that the underlying genetic loci that are encoding those ecological traits and this mating cue are actually co-localizing in the genome to some extent, as shown here on the left. So this raises the question of how the linkage between loci can affect reproductive barriers. So we already know that the linkage between loci can favor local adaptation. So for instance, if we consider this simple scenario where there are two loci that are encoding two traits, so here's the color and the size, so you see that recombination occurs between those two loci at a given rate. So here is the context of local adaptation, so that there is divergent selection on the two traits. You see in the first population, the little blue fishes are favored, while in the second population, the big red fishes are favored. We know that in this context of local adaptation, a tight linkage between the loci, that is a low recombination rate between the loci, can favor local adaptation. This is because such low recombination rates favor the maintenance of the genetic association that arise. So what we call linkage equilibrium in population genetics, so the fact that we find some alleles at those two loci more often than expected by chance. And by maintaining such strong genetic association, this increases the strength of indirect selection, so selection that occurs through genetic hitchhiking, which favor increased allelic divergence between those two populations and therefore favors local adaptation. So importantly, it has been shown that this occurs even if one of the traits is a signal that is used as a basis for associative mating. For instance, if the color is a signal so that individual may prefer to mate with individuals that share the same coloration than themselves. So it has been shown in a previous study by Savidio and Berger in 2020. We could reproduce these results in a more recent paper. So here is what's happening in a two loci population genetics model. So this time we assume that it's a haploid loci and not diploid like in the previous study. And we also assume that only females express mate choice to make it simple. And we also assume that the strength of associative mating is fixed. So we have here a model with two loci, so exactly the same scenario I just told you in the previous slide. One locus that is encoding for an ecological trait, here the size, and one locus that is encoding a signal used as a basis for associative mating, here the color. And in this figure, I represent the allelic frequencies at the two loci, depending on the value of the parameter that controls the strength of associative mating. So this is in the two populations, so the plain line and the dashed line. So you can first see that at those two loci, there is allelic divergence. So when one allele is common in one population, it is rare in the other. And you can also see that for extreme value of that parameters, there are no lines. This is because allelic divergence is lost, so those lines are not represented. So allelic divergence and therefore local adaptation is lost for extreme value of that parameter. So these lines are for a recombination rate between those two loci is equal to 0.5. So it means that those loci are loosely linked in the genome. Now I'm going to show you what is happening if the recombination rate is lower. So for 0.2, 0.01, and for 0. So first, you see that allelic divergence is maintained for a broader range of parameters. And second, you can see that allelic divergence is getting higher. You see that the plain line and the dashed line are becoming further apart, which is, in conclusion, what I just told you in the previous slide. A low recombination rate between the loci favors local adaptation, even in that case where one locus is encoding an ecological trait under selection, and the other locus is encoding a mating signal used as a basis for associative mating. So here, the linkage between loci favors local adaptation. Now, I'm going to show you what is happening in terms of mate choice that lead to prezygotic isolation. So during my last postdoc with Maria Saviggio, I studied what happens if the strength of associative mating can evolve. I investigated how the genetic linkage can affect prezygotic isolation caused by mate choice. So again, I formulated a verbal hypothesis. So I told you that magic traits are traditionally thought to strongly promote reproductive isolation, yet a very tight linkage between loci mimic to some extent the case of a magic traits, because a single cluster of genetic loci would encode both the signal and the ecological trait under selection. And therefore, I hypothesized that a tight linkage between loci would favor the evolution of such mates. To evaluate this hypothesis, I considered the scenario I just told you about with local adaptation with divergent selection on one ecological trait. Also, I assume that there is a signal used as a basis for associative mating. So each of those two traits are encoded by single genetic loci in a haploid model. So it's basically the same model as the one I just told you, except that I considered an additional locus which is encoding the strength of associative mate choice. And thanks to the implementation of this third locus, I could conduct an invasion analysis to infer the strength of associative mate choice at evolutionary equilibrium. So basically, the strength of associative mate choice at evolutionary equilibrium is the value of the trait that we expect to evolve given subsequent mutation at this third locus. So here is what we get. So here is the strength of mate choice at evolutionary equilibrium, depending on the recombination rate that is implemented. So this is the recombination rate between those two focal loci. And you can see here that if the recombination rate is low, only weak associative mate choice is expected to evolve. So a low recombination rate between the loci inhibits the evolution of associative mate choice, which is the opposite of the verbal hypothesis that I told you. And this we can explain if we consider the fitness landscape that we have in this model. So this is a context of local adaptation where the fitness of the recombinant genotype is particularly low here in the middle. And associative mate choice is favored because it reduces the risk of producing those maladaptive recombinant offspring. And if the recombination rate between those two loci is low, the risk of producing those maladaptive recombinants is already low, which explains why only weak associative mate choice evolves under this condition. So this line is for the case where this third locus is tightly linked to the others with a very low recombination rate. So that this situation is different if we consider a higher recombination rate between this third locus and the other loci. So we see that we lose these effects, which makes sense given that the evolution of mate choice relies on indirect selection and therefore on the genetic association that arise that depends on the recombination rate between this third locus and the other loci. So to sum up, all this relies on indirect selection on the strength of mate choice. So I'm not going to show you what is happening in terms of reproductive isolation, but we could show that for low recombination rates, when we get weak associative mate choice, we get actually an overall reduction in reproductive isolation. So to conclude on that study, so even if the linkage between loci favors local adaptation, it inhibits the evolution of mate choice, leading to an overall reduction in reproductive isolation, which I find particularly interesting because, as I told you, magic traits are thought to strongly promote reproductive isolation, and yet here, when there's a tight linkage between loci, which mimic the case of a magic trait, we get the opposite outcome. So this is overall some kind of negative coupling that is driven by the antagonistic impact of genetic linkage on the reproductive barriers. And to finish, I'm going to show you how a third mechanism by which negative coupling can occur. I will show you how sexual selection can lead to negative coupling. So this can occur when mate choice leading to prezygotic isolation is based on multiple signals, and I'm going to show you the case where it is based on 2 signals. So in nature, in animals, so mating signals are usually multimodal. So those are usually combination of acoustic and visual signals. So for instance, in this illustration, you can see that the courtship display of the Drosophila is actually rather complex, and the signals are actually both acoustic and visuals. So if we put it simply, if we assume that there are only 2 signals, here the color and the size, as represented here on the left, you see that if we assume that each of those signals are encoded by single genetic loci, there are 4 possible combinations of signals. And in this condition, females may express preference for specific combination of signals. So for instance, this female would strongly prefer big blue males, as represented by this big blue heart, and she really dislikes little red males. This female here strongly prefers little blue males, and so on. So with this condition, you see that first, female preference can actually lead to variation in male mating success. So it can impose sexual selection on the signal through variation in male mating success. And also, you can see that depending on the signals that are expressed by females, you may have situation with associative mating. For instance, as represented here, in the case where the female expressing preference for specific combination of signal also express themselves this specific combination of signals. So this depends on the genetic association that arises between those loci encoding the signal and the preference. So during my last postdoc, I also studied what happens if associative mating can be mediated by such multiple signals and their associated preference. And I hypothesized that it could lead to strong reproductive isolation. So especially in the context of local adaptation, where there is divergent selection in the signal. So if we assume that the signal is a magic trait, in this condition, mate choice is expected to lead to the establishment of positive genetic association, leading to divergent selection on the preference through indirect selection. But basically, we can expect positive genetic association to arise, leading to this condition I told you, with strong associative mating, therefore strong reproductive isolation. To evaluate this hypothesis, I built a 4 loci population genetic models where 2 loci are encoding the signals, here the color and the size, and 2 loci are encoding the associated preferences, so color-based preference and size-based preference. So I consider the scenario of local adaptation so that there is divergent selection on the signals. So with this model, I could track the linkage equilibrium, especially when the stationary state is reached. So the linkage equilibrium in population genetics, it's just a genetic association that arises. So the fact that we find some alleles together more often than expected by chance. And I could also measure reproductive isolation, and I compared reproductive isolation between the case with only one of those two preference loci versus the case where the two preference loci are present in order to investigate what is happening when the two prezygotic isolating barriers coincide. So here in this result, on the axis, I'm varying some parameter values that are actually the recombination rates. So you don't need to remember that those are recombination rates, just remember that those are parameter values that I'm changing. And I'm going to show you some colored pixels that represent the linkage equilibrium between signal loci on the left and the linkage equilibrium between preference loci on the right. So those are the genetic associations that are actually normalized so that they are between -1 and +1. And given the verbal hypothesis that I told you, I was expected that this figure would be all red, that only positive genetic association would arise. So here is what we get. So first you see that we get an outcome that is rather complex, even if this model is extremely simple and with only 4 loci, we get some abrupt changes in the model outcome depending on the parameter values. But also you can see that there is some blue in those figures, which means that we have negative linkage equilibrium that arise and that becomes established. By negative linkage equilibrium, I mean that the recombinant genotypes become more common than expected by chance. And those recombinant genotypes, for instance, at the signal loci, are those combination of signals that are not favored in either population, and yet they become common at the stationary state. This is because of sexual selection. This is because those recombinant genotypes at the signal loci benefit from a high mating success. And this, again, is because their frequencies of those recombinant genotype at the signal loci is much lower than that of the recombinant genotype at the preference loci that strongly prefer them. So basically, sexual selection leads to the establishment of negative linkage equilibrium, which is the opposite of the verbal hypothesis that I told you. Here is what's happening in terms of reproductive isolation. You can see that here, depending on those same parameter values than before, the change in reproductive isolation when this second preference locus is included. And you can see basically that it's mostly blue in this figure, which means that reproductive isolation is often lower when mate choice is based on multiple signals. So this result is explained again by sexual selection. So first, because of this negative linkage equilibrium I just showed you. So given that those maladaptive recombinant genotypes that are not favored in either population are more common than expected by chance, this reduces allelic divergence between the population, and therefore it reduces local adaptation. And it is also because sexual selection by itself reduces local adaptation. So it has been shown in a previous paper in a much more simple model than that, but it also occurs in the model. So basically, to conclude on that study, that last study, I showed you how sexual selection can also lead to the negative coupling between reproductive barriers. So this occurs when mate choice leading to prezygotic isolation is based on multiple signals. So let's conclude on all this. So what should we remember from all this? So I showed you in those 3 studies that the evolution of mate choice can lead to negative coupling in one way or the other. So due to the evolution of mate choice, reproductive barriers may operate together in such a way that reproductive isolation is actually reduced and not increased. In my opinion, this raises some open questions that could serve as avenue for future research. So first, does negative coupling occurs when other barriers are involved, in particular post-zygotic barriers, or does it occur mostly when there is such evolution of mate choice? So this could be investigated further using proof of concept model. And also, more importantly, we are in need for empirical evidence of negative coupling. So I appreciate that this could prove tedious, and perhaps the simplest way to begin with would be to try to find some indirect evidence of negative coupling, for instance, by precisely measuring the mating success of hybrids, given that in the last study I showed you that through sexual selection, it could lead to negative coupling. So overall, I showed you that reproductive barriers do not necessarily reinforce each other. The opposite outcome can occur. Negative coupling can occur through various mechanisms. And this, in my opinion, can have some broader implication in evolutionary biology, in particular on the link between microevolution and macroevolution. So there is a lot of work that is currently devoted to investigate how genetic and phenotypic diversification at the microevolutionary scale translates into species diversification at the macroevolutionary scale. I believe that if negative coupling occurs in nature, that it could make this investigation even more difficult than what it is already. If negative coupling occurs, it means that the establishment of reproductive barriers will not necessarily increase the likelihood of speciation because it could inhibit the evolution of other barriers. And perhaps this is something that should be taken into account in the future to tackle this important question in evolution biology. So to finish, I would like to acknowledge all the co-authors that contributed to these 3 studies. I would like in particular to thank Maria Savedio, with whom I did my last postdoc and with whom I learned a lot in population genetics theory. And thank you for your attention, and I'm happy to take your question.
Thank you very much for this interesting studies. Are there any questions?
Well, maybe a rather open question, but so it might seem that positive interaction between barriers is, well, naively thinking it might be more common than what you showed with negative coupling. So do you have any idea how many barriers actually there would exist between species with different strengths? How that could influence, like what would be the scale of the negative coupling you discovered? Would be just a tiny bit of variation, and things like that.
Yeah. So yeah, this is an open question. So I will not answer your question like in a precise manner, but basically there are some meta-analyses that start to quantify the strengths of reproductive barriers, in particular in plants. So we know that in those cases, at least of undergoing speciation, there are multiple barriers that are occurring at the same time. So both prezygotic and postzygotic barriers. Then to what extent positive coupling or negative coupling occur? I think at that stage, we don't know the answer to that question. So basically, I can't answer your question.
In your models, did you consider or allow for cases where you could have disassociative mating?
Okay, so in in the 2 last studies, I didn't. So it's only positive. But I believe that, given that we showed that a non-zero positive associative mating is favored, that disassociative mating would be disfavored, but I'm not 100% sure. There could be multiple equilibria. In the first study, I think we didn't neither consider the disassociative mating, but given again that this is a case where there is disruptive selection which favor positive associative mating, I believe that the opposite outcome would not evolve.
Does having strong, oh, this is working. Does having stronger selection make it more likely or less likely, stronger divergent selection, make it more likely or less likely that you'll get the negative coupling?
Oh, okay. So the impact of the strength of variability selection, which is divergent. Okay, so we did vary the strength of variability selection. So if I remember correctly, in the last study, for instance, we had other issue when selection is weak. I mean, we lose divergence a lot. So, I mean, we had other outcomes that made the comparison difficult. In the first two studies, if I remember correctly, stronger variability selection, which is either divergent or disruptive, favors the evolution of strong associative mating. So in terms of negative coupling, I think it reduces the likelihood of negative coupling.
Is it? Do you think for the evolution of sexual determinism and genetic sexual last part, parameters about recombination between the signal and the choosiness and the ecological trait? We know some stuff happen with recombination and sex chromosomes.
So yeah, again, I'm not sure I can precisely answer your question. So the thing is that in that last study, we assume that there is some dimorphism. There is at least preference is expressed only in females, while the signal is expressed in the two sexes. So perhaps those are an assumption that we could have relaxed. We didn't. Perhaps this could have given us some insights about this determinism, but I'm not sure.
That's a question.
They attempt the nation itself evolve in these models.
So you mean the number of crossover? Yeah. So do you mean the evolution of recombination? Yeah.
Can evolve.
Yeah. So in those studies, it is not the case, but those are. I mean, with Marian Savello, we are working on the evolution of recombination between those loci. I mean, the one I showed you here, and basically, for instance, we could show that low recombination is favored between the loci encoding the ecological traits and the loci encoding the signals. But what we found interesting is that we could show that the loci encoding, the recombination rate between the preference loci and the signal loci is completely neutral and does not evolve. And this is something we are working on right now.
I found the dynamics of the simulation that you showed in the first example very intriguing. I think I understood what happens, but I don't understand why it happens. I was wondering whether you could comment upon them.
Okay. Why we get those coevolution dynamics? So I mean,
Well,
the reason why.
I would be even more specific. Can you show the slide?
Yeah, sure.
Make more specific questions.
Yeah. Okay, tell me which.
The one with the dynamics.
Yeah. Okay. This one.
Yeah. So I think there is a first where choosiness of females is neutral.
Yeah.
While choosiness of males is fixed.
Yeah.
So in that part, I don't understand why only drift is expressed only in choosiness of females.
Yeah. Okay. I can.
We go to.
Yeah. I can first.
Okay.
I can first answer this question. So basically, there is weak selection on the female choosiness when males are choosy. So that's why there was like big deviation on that axis. And this is because when males express assortative mate choice, there is no longer a benefit associated with female assortative mate choice, because even the females that don't express mate choice mate only with matching males.
And so why the reverse is not true? Or rather
So
the
this is
converse?
because of the assumptions that we made, like the differences between males and females. And in the case of the evolution, so there is, okay, another thing, so that why there is this triggering of coevolution dynamic is because there is a threshold of the frequency of choosy female below which the direction of selection on male choosiness changes. So above this threshold, male choosiness is favored, while below this threshold, male choosiness is disfavored. And this is okay. This is because it's kind of the same explanation. If females are choosy about whom to mate with, let me think again. It's been a while. Okay, we are in a condition where male choice is kind of costly, so it's for the value. So if females are choosy about whom to mate with, no males pay the cost because they only encounter female that are matching females. If females are not choosy about whom to mate with, then males may risk to court unpreferred females, and therefore they will pay the cost, and that's why male choice is disfavored. I don't know if it's clear enough.
I don't know.
So not clear
I'm
enough.
not convinced, but I don't want to monopolize it.
Okay, we can talk about it later.
I was wondering if the order in which the barriers might evolve makes a difference. So is there an influence of the sequence of evolution, and if one barrier evolves once the other one is almost fixed, for example, does it make a difference?
I think that it could make so we didn't investigate it. We didn't investigate that in detail, but I think it makes a difference because, for instance, if you assume that mate choice evolved because of indirect disruptive selection, then perhaps it requires some polyzygotic isolation, some genetic incompatibility to evolve first, so that mate choice evolves. So I think, yeah, the order can make a difference. I don't know if it answers. Yeah. Okay.
I'm not sure I understood everything, but in the second model, you model mate choice through one single locus expressing the strength of mate choice, right? In the second model,
Yeah.
mate choice is modeled just through a model reflecting the strength of mate choice,
Yeah.
right? And generally, correct me if I'm wrong, the mate choice is rather modeled through a male and a female trait that will interact by Bayesian paternities, right? So
Yeah.
do you expect your results to be affected if you would have a male and a female trait in your second model, and for instance, the male trait being linked to the magic trait?
Yeah. Okay. Yeah. So in the second study, we did consider that assortative mating is taken for granted. Like, it doesn't rely on preference loci that determine the direction of the preference. And for sure, I mean, this would change the results because in the second case, I mean, some genetic association need to arise, and I mean, we would probably have different results, but we didn't look at it further because it's very quickly a mess when we increase the number of loci. So.
Okay, well, let's thank Thomas again. Thank you.